So polyadenylation is the final RNA processing event after transcription, and this polyadenylation is of mRNA is intimately linked with the termination of transcription, although exactly how is still not quite clear. So when the polyadenylation of newly synthesized mRNA takes place, it causes termination of transcription. Recently, however, an enzyme that degrades the second RNA as it emerges from the polymerase has been identified, and this enzyme may itself trigger termination. This is called the torpedo model of termination. The free end of the second RNA is uncapped and thus can be distinguished from genuine transcript. So one is the genuine transcript which has capped ends, and the other one which is still emerging out from the polymerase, that is uncapped. So these enzymes, RNases, can easily identify these uncapped RNA. This new RNA is recognized by an RNase called RAT1 in yeasts and XRN2 in humans, that is loaded onto the end of the RNA by another protein, RTT103, that binds the CTD of RNA polymerase. So this RTT103 is bound to the CDT tail of polymerase, and it causes these RAT1 or XRN2 to bind to the newly synthesized RNA. The RAT1 enzyme is very processive and quickly degrades the RNA in a 5' to 3' direction until it catches up to the still transcribing polymerase from which the RNA is being spewed. So it rapidly degrades the RNA which is extending out from the polymerase, and it reaches the that site from where the newly synthesized RNA is coming out from the polymerase. Termination may not require very specific interaction between RAT1 and polymerase, and might in fact be triggered in a manner rather similar to that of RHO dependent termination in case of bacterial transcription. The highly processing RNase polymerase either pushes polymerase forward and or pulls the remains of the nascent RNA transcript from the enzyme. So in two ways it can stop the transcription. So it can either pushes the polymerase forward, or in other case it can pull the nascent RNA from this machinery. So in both the cases transcription will terminate. It is also possible that other factors are needed in addition to RAT1 to dislodge polymerase, as in vitro RAT1 is alone insufficient to carry out this function even after it has degraded the transcript. So even if the transcript RNA has been degraded, it is not possible to terminate this if only RAT1 is available. So this is the picture you can see. This is newly synthesized RNA, and after this polyadenylation it is cut, and this is the second RNA, and this RAT1, then it started hydrolyzing this RNA strand, and finally it reaches here and it causes the termination of transcription. Although the torpedo model for termination is now the favored one, there is an alternative model called the allosteric model. According to this model, termination depends on a conformational change in the elongating polymerase that reduces the processivity of the enzyme, leading to spontaneous termination soon afterward. So there is some mechanism which causes the polymerase, elongating polymerase change, conformational change in the elongating polymerase, and this change then causes the termination of transcription. This conformational change would be linked to polyadenylation and could be triggered by the transfer of 3' processing enzymes from the CTD tail of polymerase to the RNA. So here you can see this is the second model, and according to this enzyme will cut it, and then this polymerase, which is here, you can see the shape, and here you can see this is a conformational change in the polymerase. It is oval here, and now it has become a bit polygonal. So this conformational change results in the termination of transcription.